let's go ahead and get started here. So as I mentioned just a little bit ago, we're fortunate to have the team from Heidelberg, Germany here, with the exception of the one in the middle there. But here with us today is Rachel Gilmore. So Rachel, I mentioned, is on a 3-year grant from FPWR as a postdoctoral PhD fellow in Christian's lab. We also have Tim, who's been in the lab for quite some time. Felix, who I met for the first time. Maybe Felix, you can share some hair with Ferdinand. By the time, what's your thoughts on and Ferdinand here with us as well to give us an update of what's going on in Heidelberg, including in Dr. Schaaf's lab and in Ferdinand's lab at this point. So they are here as a resource for you. So make sure you ask questions, right? It's a small enough group that we can be very conversational along the way. Also, this is on Zoom and it's being recorded at this point. So if we have folks that want to listen, grandparents, friends, or other folks with Schaaf-Yang syndrome, we can do that as well. So without further ado, I'll turn it over to the team.
And that's great. Okay, thanks so much, Matt. Good morning. We're really, really excited to be here today. Thank you for letting us talk to you. We're speaking on behalf of the Schaaf and Alzheimer labs from the Institute of Human Genetics in Heidelberg. And we have Schaaf-Yang syndrome as one of the core themes that we investigate in our research there. And today is our chance to give you something back, basically an honest snapshot of what is happening in research right now, what we know about Schaaf-Yang syndrome, what we know about MAGEL2. Now, research moves carefully and sometimes takes quite some time, so we will not have all the answers that you deserve, but we'll try to give you as many as possible. And we do have new data to share, new ideas, and a growing sense of momentum that we're moving into really understanding pathomechanisms and investigating potential therapies. Now, before we get into the details, I just want to repeat what Matt just said. Please feel free to just put your hand up at any time if anything's unclear, if I talk too fast, or if you want something explained better. And there will also be time for questions at the end, but please feel free to ask as we go along. So at the beginning, just a quick recap of Schaaf-Yang syndrome and its natural history. You're probably the better experts on this than us, but what we see on average in Schaaf-Yang syndrome children is the following. In infancy, the most pronounced symptoms are usually muscular hypotonia, so weak muscle tone, contractures of the joints, which is also something that is unique to Schaaf-Yang compared to Prader-Willi syndrome. We see respiratory distress and feeding difficulties. In childhood, what is more pronounced are intellectual disability and developmental delay phenotypes. And in quite a lot of children, we see autism spectrum disorder. And this is also something that stands out compared to Prader-Willi syndrome, where this is rather, it's not rare, but it's less common. In the few adult patients that we know of, we see obesity phenotypes, even though they are not as pronounced as in Prader-Willi syndrome. And we also see an increased prevalence of psychiatric conditions. So this is what we see on a phenotypic level in terms of symptoms, but Schaaf-Yang syndrome is inherently a genetic condition, and maybe this is too easy for some of you, but I will just go over the genetics once to have everyone on board. So when we're born, we're basically just, in the beginning, we're a lump of cells, and then we develop gradually. But still, our bodies just consist of many, many cells. And within each of those cells, the cell carries the DNA. And DNA is basically a blueprint or a big library that has all the building instructions for our entire body. And each cell has the same DNA within it. Now, DNA, when we say the whole DNA is sort of the library, the individual books are chromosomes. So DNA is packaged into chromosomes, and we have each of those books twice. we get one copy from our mother and one copy from our father. Now, when we zoom in more into the chromosomes, what we see at the more micro level is that DNA consists mostly of genes, and a gene is a functional element within the DNA, and genes are the elements of DNA that in the end code for proteins, but we'll get to that in just a minute. Since we have each chromosome twice, we also have each gene twice. We get one of the copies from our moms and one from our dads. Now, what is special about MHL2, the gene involved in Schaaf-Yang syndrome, is that even though we have two copies, only one of them is active, which is the father's copy. At the same time, the mother's copy is silenced. So even though probably most of your loved ones have an intact copy of MHL2 on the maternal chromosome, still, if the paternal chromosome is affected, that leads to Schaaf-Yang syndrome. Any questions so far, or is that fairly clear? Okay, perfect. Now, coming back to proteins and really simple basic biology, we go from our DNA to RNA by a process called transcription. And if DNA is the library with all the books, RNA is more or less just the working copy of that. And we then can translate RNA in our cells into proteins. And proteins are like little machines that do things in our bodies. They can have lots of different functions within the bodies. And we'll talk a bit about what we know about MHL2, but actually lots of proteins have functions that we don't really fully understand yet. Okay, so we go from DNA to RNA to protein. Now, as we grow, we also need cells to divide and split because our bodies need to grow. And when cells split, they need copies of DNA. And this process of duplicating the DNA within a cell is called replication. So we replicate the DNA that we already have. And in that process, unfortunately, sometimes mistakes do happen, so-called mutations or variants. You can think of that as typos within the genetic code. And when these variants occur in DNA, this process of transcription and translation is different. So going from a DNA with typo to RNA can sometimes mean that no RNA is produced at all, and then no protein can be translated. But sometimes it also means that RNA is being produced that just looks different to the RNA that you would normally get with that gene. And this is what happens in most cases of Schaaf-Yang syndrome. So we get an RNA product, but it just looks different. And this different RNA also leads to a protein that looks different to the normal MHL2 protein. We can also look at this specifically for MHL2. So again, MHL2, I've already mentioned that name a couple of times, is the gene that is involved and affected in Schaaf-Yang syndrome. This is a gene that codes for a protein of the exact same name. And again, it's only active on the paternal copy of the chromosomes. Now, you can see here a comparison of what the gene looks like in healthy individuals. And in individuals with PWS, this gene is completely deleted, so they don't have that copy anymore. In individuals with Schaaf-Yang syndrome, on the other hand, we don't have a complete deletion. What we do have are variants that can be basically anywhere within the gene. There's some parts of the gene that are more prone to be affected, but they can basically be spread. And what that means on a protein level for these three comparisons is normally we just get the normal MHL2 protein. In PWS patients, what we see is they have no MHL2 protein at all. But in Schaaf-Yang patients, what we see is that they get part of the MHL2 protein, but it's shorter And sometimes it also has parts of it that are not part of the normal MHL2 at all, but look completely different, which is indicated by this orange box here. So this is a really important concept to understand. This may make the difference between Schaaf-Yang syndrome and Prader-Willi syndrome, because in Prader-Willi syndrome, we see no protein, whereas in Schaaf-Yang syndrome, we see this altered protein. Now, how do we learn more about the function of MHL2 and the mechanisms that underlie Schaaf-Yang syndrome? For that, we'll take you into the lab at Heidelberg and tell you a bit about the kind of research we do there. We basically have three main model systems that we use. We have cells that can be derived from patients and where we can sort of model the types of variants that we see in patients. We have MHL2 deletion mice, which don't have the MHL2 protein, so they are more similar to Prader-Willi syndrome patients. And then we have this new rat model that was recently generated, which has a truncating variant, which means we get a shorter protein in the rat that is somewhat similar to what Schaaf-Yang patients usually have. And most of what I personally do is focused on this rat model. Felix is also going to talk a lot about the rat model and why we think that it's a really good model for Schaaf-Yang syndrome, or at least the best model we currently have, even though it's not perfect. So what we've learned from the rats so far in terms of basic pathomechanisms is the following. So first of all, we know that Schaaf-Yang syndrome presents with more severe phenotypes compared to Prader-Willi syndrome. Children have more severe intellectual disability on average, longer developmental delays, and we also have some symptoms that we don't see in Prader-Willi syndrome at all. There are also some individuals that have complete MHL2 deletions that completely lack the gene, which don't get Schaaf-Yang syndrome. So we really think that this altered protein has some sort of negative effect on the cells. And what we do know from a group in Spain is that, think of the green thing circle there as the normal sequenced MHL2 protein and the red one as the shortened version. We see that this shortened version shifts its subcellular localization. So it goes from being in one part of the cell to a different part of the cell. And this may be part of the mechanism why it has a negative effect on cellular function. And Rachel is going to talk more about these potential effects and how we could maybe treat them. Now, this is a very busy slide, but bear with me. I'm going to explain that diagram in a lot of detail such that everyone can understand. So what we're looking at here are basically all the proteins that we see in the brains of our MHL2 p-mute rats. And we're comparing the proteins that these rats have to the proteins that their littermates have, which are rats that are not affected by MHL2 mutation. And the further we go to the right, the more a certain protein is increased. And the further we go to the left, the more it is decreased. And the further we go to the top, the more significant the change is. So you can see the one at the very top is oxytocin. That's that little peptide here. And if we go to the right, that means it is upregulated. So what we find in our MHL2 rat model of Schaaf-Yang syndrome is that these rats have far more oxytocin compared to rats that don't have the MHL2 variant. Oxytocin is a hormone that has lots of different functions during brain development and in social behavior. And it is it was very confusing to us that we would find more oxytocin in the rat model of Schaaf-Yang syndrome instead of less. So the question is, does that make any sense? And I'm going to walk you through why we think that this finding makes sense, but why we think that oxytocin may still play a role in pathophysiology. First, as a little mental break. This is a nice image from our studies of the rat brain. This is a slice of hypothalamus, and all the red things that you can see are oxytocin cells that we stained with a red dye. And in this three-dimensional image, we have lots of those. We can label each of the cells with a little sphere. And what that allows us to do in quite a cool computational program is that we can count all the cells that the rats have. And we can also look at how bright the cells shine. And the brighter they are, the more oxytocin they contain. We did this for lots and lots of rats, and we did this for lots of rats that have the MAGE2 variant and compared them to the rats that don't have the MAGE2 variant. And what we see are two things. Don't even have to really look at the graphs. What you see is just, we see more oxytocin neurons, and we see higher oxytocin content within the cells. Sorry, was that a question?
I don't think so.
Okay, cool. Yes. Yes, the Schaaf-Yang rat model. So again, that's a bit counterintuitive, right? Yes. Okay. Yeah, then we were confused as well. So yeah, keen observation. And we're going to get into why we think that happens. So again, we see more oxytocin neurons in the rat model, and these neurons seem to contain more oxytocin within them. Yeah, actually, that's more or less what we think happens, and I'll show you in just a minute. So what we see is the following. This is the kind of data that we look at when we look at proteins. So each of the dots, each of the little dots is a protein. And the bridges between proteins means that these two proteins, for example, do something together. They're somehow involved in the same cellular pathways. They somehow have similar functions, or they build little machines together. And I think if you look at this graph, what you notice is that there's this lump of proteins down there that all seem to do something together. And now the question is, what does this lump do? Here's a very simplified model of a cell with only the structures that we're interested in. So when oxytocin is being produced within the cell, it needs to go into these little ball structures, which we call vesicles. It needs to go into these vesicles because only when oxytocin is inside the vesicles, it can then be secreted outside the cell. This is a very important process in any cell that deals with hormones and releases hormones. And then once the hormone is released, it can take some effect. Have an effect on other cells, have an effect on behavior and other things. Now, what we think happens and what this little number of cells that is dysregulated is involved in is this process of getting oxytocin into the vesicles. So this process of getting oxytocin into the vesicles seems to be impaired, which means oxytocin may just be trapped within the cell, and then we cannot release it. That will also mean that it cannot take its intended effect. Again, this is just a hypothesis, and we're still following that up, but it could explain why we see more oxytocin and why we see more oxytocin cells, just because oxytocin is trapped within them. Now, if that actually happens, what that would mean is... Sorry.
Yeah.
Oh, yes, please.
So in the accumulation of the oxytocin within the cell that's unable to be released and have an effect, does it decay or does it build up? And does that cause a problem?
So we don't really know yet, to be honest. Oxytocin itself doesn't have a very long half-life, so it doesn't survive very long. It's usually degraded quite quickly. And going out on a limb. I think that's also what happens, but just because it is still trapped on average, there's still more on average.
Good.
Good question.
Very
Yeah.
good question.
So if oxytocin is not properly released from the cells, it can likely not do its job, even though it may be present within the cell. And if there is this functional deficit, right, it cannot take its effect, then there may still be a potential role for oxytocin replacement therapy, because we want oxytocin to take this effect. And if it just stays within the cells, we don't really win anything. So what is next for us is we are following these studies up with more detailed observations of the cell, where we can look at the different structures and exactly where oxytocin is located within the cells. We're also looking at different cell types and how they may be affected in the major rat model. And we really want to understand the functional relevance of our findings. For example, by looking at how well these cells can secrete oxytocin. Ultimately, our goal is that we understand the basic mechanisms that lead to Schaaf-Yang syndrome better, because that will also give us a much better insight into the condition that we're trying to treat. Do you have any questions so far on these basic pathomechanisms?
Actually, I have one
Yeah.
question, Tim. You mentioned a couple slides earlier that patients without the MAGEL2 gene are less severe than.
Yes.
How many patients are you aware of that have only a MAGEL2 mutation? It's not Prader-Willi.
So there's a few, I think, like, I can think of 3, 4, 5.
Less than 5. I
Yeah,
think.
something around that.
And they're all neurotypical?
Then I don't think they're completely neurotypical, but they don't develop the Schaaf-Yang phenotype. They're closer to the Prader-Willi phenotype, but they're far less affected.
Super mild. Like, yeah.
Yeah.
And
Think
it's not just typically just a MAGEL2 deletion. Usually there's a couple genes, but it's a much smaller deletion than what we see in
Yeah.
Prader-Willi.
Yes.
Will you be connecting this research with the use of oxytocin, like, and how that symptoms like potentially addresses some of these issues?
Yes. So what the research that I've talked about up till now does is basically tell us something about what happens when MAGEL2 is changed. But what my colleagues are mostly going to talk about is how can we address these changes and treat that. Yes.
So if their body isn't processing oxytocin correctly, and then we're giving them an oxytocin replacement, like the nasal spray, then how are they processing that differently? Like, why are we, would their body like still not process it? You know what I'm saying? Like, what's the difference between?
Yeah, of course. Yeah, I will go into that again. So normally what happens in the body is you have oxytocin within the cells, and then it's pushed out of the cell. And when it's out of the cell, it can have its effect. Now, what we would do with oxytocin replacement therapy is, or what we're trying to do with it, it's hard to actually get it to where it's supposed to be. But what we're trying to do is get oxytocin into the room outside the cells. So we're not pushing it into the cells, but it goes outside. And therefore, we wouldn't have that problem with the cellular mechanisms being disrupted, because it would be outside the cells. Yeah.
Yeah, we're basically skipping the broken bridge. Gotcha. We're just putting it immediately where it needs
Is
to
that
go.
kind of how the growth hormone replacement works?
Yes,
It's doing like you're getting that, so it's not front dress cells, it's
exactly.
getting right
Yeah.
into the body.
And they can still process it downstream.
Yeah, because it's not going through the cell and getting caught, skipping that.
Yes.
Thank you guys for this. It's amazing. I just wanted to kind of share my experience with carbitocin in PWS. Anecdotally, I noticed that the younger patients, all injured, And that would follow, I mean, MAGL and NDN in PWS, but MAGL is responsible for transporting hormones outside of the cell. And if it's not there or it's not functioning well, it's going to cause accumulation. That makes sense. The problem then is that over time there'll be desensitization of cells outside. Receptors will basically, if you don't use it, you lose it, would be the concern. And perhaps that's why carbetocin is working so much better in the pretty, young kids. So, and that's why there's a lot of promise in PWS with early use of oxytocin. So is that like considered, because I want to help the SYF community now. And my question is, how early, like, do the clinicians, do all your clinicians know to start oxytocin, like, really early? Is there evidence to support that use? And oxytocin is extremely short acting. Has carbetocin been considered in SYS?
So I know that there's, thank you for that. There has, compared to Prader-Willi syndrome particularly, we don't have any clinical trials of oxytocin therapy in Schaaf-Yang syndrome yet. We know that there's a couple of clinicians who prescribe oxytocin therapy, but it has not been scientifically evaluated, so I don't feel comfortable making a statement on that. I think that what I can say from the studies that we've done is the rats seem to be particularly affected in earlier developmental stages. And what we see in trials in ASD and Prader-Willi syndrome is also that oxytocin seems to have its effect dependent on the developmental stage and the context where therapy is given, maybe even in combination with behavioral therapy. But we don't have any evidence for Schaaf-Yang syndrome yet.
There might have another question. Can you go back into the slide where you showed oxytocin stuff regulated stuff, right?
This one here?
Yeah.
Yes.
Has there been any research done on this downregulated protein?
This one on the left.
That or even further on the left?
Yeah, so there's only one that stood out here, which is this one. And no, we haven't looked into that further. Most of the proteins that are somehow dysregulated are somehow involved into how proteins are being produced or how cells communicate with each other. But so far we focused on oxytocin because it's the one that stands out the most. Yes.
Regarding growth hormone, there have been thinking done, like, once the bone are not ready to close, so the growth plates, right, are closing, to what amount of growth hormone that they should still continue with. So just throwing it out there, like, we just were with endocrinology and like growth plates are about closed, so then there's an adult dosage, and that's more for metabolism.
Yeah, that's
like if there's a benefit or what that magic formula is as the kids grow, if they need more than just a typical kid.
Yeah, that's a great question. So what we know about growth hormone in Schaaf-Yang syndrome is mostly from a retrospective study, which means we didn't find individuals and said, okay, part of them are going to get growth hormone and the others are not, and we compared it to groups. But what we looked at the institute a couple of years back was children that received growth hormone therapy and how did it help them. I don't think we have enough data to really give a good estimate of good levels of growth hormone, unless anyone wants to disagree with me on that. So I think that in this case, usually the endocrinologists actually have more expertise than us. What we can say is more that growth hormone in general seems to be an interesting treatment to pursue, but the actual treatment is usually also in the clinic not done by geneticists, but by the endocrinologists who are the specialists focusing on hormones. So they will know best.
Jennifer Miller,
Yeah.
I'm big in question.
That's what I was like.
Yeah. Okay. Now we also have like But first of all, I want to say some of this was based on hypotheses, and we're still in the process of validating it. What I also want to say is there's lots more people than me behind this kind of research, and they will be acknowledged on the last slide as well. But now I would really like to hand over to my colleague Felix, who's going to tell you more about the behavior that we see in the rat model.
So thank you very much, Tim, and a warm welcome to everyone also from my side. It's wonderful that you all made the way here to Phoenix. Yeah, thank you all for coming, and thank you for being the biggest inspiration for our research that we have. My name is Felix, and I'm also a doctoral student at the labs at the Alzheimer and Schaaf lab in Heidelberg. And I'm very excited to also share some of the research that I have performed on Schaaf-Yang syndrome within the last year. And so Tim has already mentioned that we have different ways in the lab to find out more about Schaaf-Yang syndrome. And one particularly important approach is the use of animal models. And while there has been work on several animal models focusing on major 2 in the past, I think at this point we can say that one animal model especially stands out as being suitable to study Schaaf-Yang syndrome. And this is a rat model that we refer to as major 2 p-mute rats. And unlike previous models that completely lacked major 2, this one actually carries a specific variant on the paternal copy of the gene, which results in an altered protein. And this is actually very similar to what is seen in individuals with Schaaf-Yang syndrome. So what I'm basically saying is that the genetic background of this model is much closer to what we see in humans than previous models. So we think it's much better for modeling the condition, and also much better, potentially much better for looking for treatments. But before I will talk about how we potentially could use this model to evaluate treatments, first of all, we have to understand what features of Schaaf-Yang syndrome are actually replicated in this rat model. Because a good model should not only have a genetic change that is similar to what is seen in individuals with Schaaf-Yang syndrome, but it should also reflect some of the symptoms that we see in humans. And so I really spent a lot of time with these rats and studied their traits and their behaviors. And the very first thing that I noticed was regarding their weight. And on this graph, you can see the weight development, and you can see two curves, and you can see the weight of the rat model of Schaaf-Yang syndrome in red, and the weight of the brothers and sisters that do not carry the variant in major 2 in blue. And what I think is really visible is that at all time points we had a look at the Schaaf-Yang syndrome rat model weighs less than the brothers and sisters that do not carry the major 2 variant. And this is quite interesting, as it may mirror the growth deficiency that we often see in Schaaf-Yang syndrome. And following up on this reduced body weight, we decided to also have a look at the eating behavior in this rat. And this was quite interesting. Again, you can see on this graph in blue the siblings of the rats that do not carry the variant in major 2, and in red you can see the rats with the variant, so the Schaaf-Yang syndrome rat model. And what we are having a look at is the eating duration of these animals within 24 hours. And interestingly, they actually spend more time eating despite having reduced body weight. And we think this is because they just take longer to eat their food and actually mirror some of the feeding difficulties that we also see in the condition. But we did not only want to have a look at feeding behavior and at weight, but also at some social aspects in the rat model. And the very first thing I had a look at was their way to communicate. And some of you parents may remember that maybe your loved one with Schaaf-Yang syndrome at some age has shown atypical crying or just challenges to express their emotions. And we wanted to see if we see something similar in the rat model. So what we did in this experiment was to isolate rats at very young age for a short period of time for their mother, and then record how they call for their mother to be picked up again. And yeah, this is often compared to the human behavior when a child is removed from their mother or their parents and then cries for them to be comforted. And what we did was to analyze these calls that were emitted from the rats. And actually, we found that they looked quite different from the ones that we see in the littermates that do not carry the variant in major 2, which may mirror some of the challenges in communication that we see in Schaaf-Yang syndrome. But also, we wanted to have a look at how these rats directly interact with each other. And I have now talked a lot about their behavior in this rat model, and I think it can sometimes be quite hard to picture how these experiments actually look like. So to better visualize this, I have brought a little video which should show how altered social behavior can look like in rats. And what I would like you to have a look at is the green rat and compare it to the behavior that the red rat shows. Okay, and what I think is quite visible is that while the red rat tries to socially engage with the green one is much more interested in the environment and actually somehow tries to avoid the social interaction. And while this is not fully representative of what we see in the rat model, we have done a very detailed video analysis of their behavior and have found that in fact the rat model shows reduction of certain social behaviors and also shows an increased exploration of the environment, which may mirror some of the alterations that we see in social behavior in Schaaf-Yang syndrome. Okay, and now you may ask, why am I telling you all this and why are we having a look at all these changes in the rat model? And the reason is that we want to make use of these changes in the model to evaluate potential treatments for Schaaf-Yang syndrome. So we have these changes such as reduced weight or abnormal feeding behavior. Then we want to give a treatment to these rats and see if we can see improvements in these phenotypes of weight and behavior. And this is a great chance for us to test the effect or the efficacy of different therapies in a model of Schaaf-Yang syndrome, which then might give us a hint towards if this would also be a suitable treatment for treatment of individuals with Schaaf-Yang syndrome. And now the very first therapeutic study that we are conducting in this rat model is oxytocin treatment. And Tim has already talked a lot about oxytocin and why it might play an important role in the pathophysiology of Schaaf-Yang syndrome. So we think we have quite a good rationale for the study. And this is currently ongoing experiments in which we actually treat these rats with oxytocin at an early developmental age and see if oxytocin can improve behavioral changes and weight development in this model. But actually, oxytocin is not the only treatment approach that we are currently investigating. And at this point, I would like to hand over to Rachel, who will tell you more about another approach that we are having a look at.
All right, I'm aware of the time, so I'll try to be brief and make sure that we all get a chance to talk to you guys. So hi, I'm Rachel. I'm a postdoc in the Schaaf lab, and I have the absolute privilege of being here today to talk to you guys a little bit about my work investigating antisense oligonucleotides. And so first, I want to revisit the central dogma that Tim talked about in the beginning. And I think a lot of us typically think of DNA as this little twisted structure, this twisted ladder right here. But what I want to talk a little bit about is that each of these ladder rungs are actually pairs of nucleotides or bases. And so you have a strand from the blue strand and a strand of the red strand, and each of those has a base that pairs together. And what happens is there's a protein in our cells that actually takes this DNA and transcribes or reads it into RNA. And RNA is only composed of a single strand, so half of the ladder. And RNA also is composed of bases, though it has a little bit of a different version of its alphabet, as you might notice from this picture. Instead of T's, it has U's. But that's not all too important for you to remember. And there's a different protein that's responsible for taking this RNA and translating it or reading it into protein. And while a lot of the times we can think of proteins as just this ball of squiggles, what actually composes these squiggles is single amino acids, so the alphabet of proteins, if you will. And so now with that knowledge, we can talk a little bit about what antisense oligonucleotides are. So antisense oligonucleotides, or ASOs for short, are short man-made nucleotide sequences that are designed to pair with target RNA molecules. And what then, when they pair, this triggers, is it triggers those RNA molecules to be destroyed. And so in this case, I've kindly asked ChatGPT to make me a protein Pac-Man. In real life, this protein Pac-Man is actually called RNase H, and to the best of our knowledge, it doesn't actually look like Pac-Man. But if you think of it as a protein Pac-Man, that's good enough for me. And of course, if we destroy the RNA, then we can no longer make the protein. And so ASOs have actually been implemented to get rid of or change proteins of other diseases in the past, such as spinal muscular atrophy, hereditary transthyretin amyloidosis, and Duchenne muscular dystrophy, or DMD. And so this is not a new idea for us, right? Not just to get rid of MAGEL2, these things have been used for other proteins as well. And so why do we think that ASOs may be a potential therapeutic avenue for SYS? And so this is going to come back to the question that we've talked a little bit about in Tim's talk as well, in that in Prader-Willi syndrome and other case reports available, there's no MAGEL2 protein at all. And what we see in these reports is that the phenotype is less severe as well. And so our hypothesis then is that getting rid of this altered MAGEL2 RNA or protein may be able to kind of relieve some of these phenotypes that we observe. And ideally, this is like big picture ideas here, right? We wouldn't only get rid of the altered MAGEL2, but we would also supplement somehow with this full-length MAGEL2 molecule. And what we think that would be is like the gold standard or like the ultimate goal. Though I do want to be super transparent in that we are quite far from this right now. So there's no trophy for us, I'm sorry. But we are starting to investigate this idea in small steps. And so by using the same rat model that Felix just told you about, what I have done is I've actually taken cells from that rat model. And like Tim was talking about in the beginning, the rat model composed of all of these different cells, each of these cells also contains this altered MAGEL2 protein. And what I can do with these cells is then treat them with a bunch of different ASOs that were designed by a company called Ionis. And then I can ask the question if any of the ASOs that I put on these cells were able to reduce the expression of the truncated MAGEL2. And so I have some preliminary results from this ASO screen that I did. And I'm going to put a quite large graph on the screen, and it may be a little bit imposing, but it's not going to be because I'm going to walk you through it. And so this is my original ASO screen. And what you can see here, this is a representation of me asking the question of after treatment with ASOs, how much MAGEL2 is in these cells. And so our x-axis or our horizontal axis is all of the different ASOs that I used. And our y-axis or our vertical axis is the amount of MAGEL2 available in those cells. And each ASO that I used has two bars, one blue one and one orange one. And the blue one represents cells that have a full-length MAGEL2 copy, and the orange one represents cells that have the altered copy. So I just essentially tested each ASO twice. And in these types of experiments, we need a baseline or a control to compare all of our results to. And that is located here on this graph. It's called SCRAM for short, and it's actually short for the word scramble. And what this ASO does is it's just a mixed up sequence that doesn't target any gene. So in this case, we wouldn't expect any degradation of MAGEL2. And what we do with those samples is we set them to 1 or 100% MAGEL2 expression. And then we can compare all of our other samples to that sample. And so our expected outcome was that some ASOs would reduce MAGEL2 expression to like half of the control. This is what we've seen in ASOs in the past. This is what we've seen in our own lab with ASOs targeting other genes. And those bars would then be located in that region down there, about half of the control. And of course, as you can see looking at this graph, that's not quite what we observed. So our actual outcome is that a handful of ASOs very slightly reduce MAGEL2 expression, like 10 or 15%. And so while this was not the best outcome for us this first go around, we're going to persist. We're going to try this again, just as we do in science. So I'm not going to make any super big claims on ASOs right now at this point because we're still early on, but just know that I have already planned a bunch of experiments to do when I get back to try these again. And even though this is a super, super exciting avenue of research, we do still have a bunch of questions about potentially using ASOs for SYS. Things like, would ASO treatment alone be enough to yield a decrease in phenotypes? What would an improvement in phenotypes look like? At what age would this ASO treatment have to occur? How often would ASO treatment have to occur? Would ASO treatment work in children or adults with SYS? And would there be any side effects to actually taking away this altered copy? And so you may be wondering what you can do to support these efforts so that someday there's an effective treatment for your loved one with SYS. And for the ASO studies, right now, I just need your patience and support, which we so greatly appreciate. But Ferdinand is going to quickly tell you about another study that you can help with in the more near future. So thank you for the chance to talk to you.
Hi everyone, my name is Ferdinand Altheimer and it's so good to see all of you. I mean, I've met quite a few of you already and it's always nice to see you. And it warms my heart to see the development for Hayden, especially. I think I've met you the last time in Denver, so it's really nice. And I want to take this opportunity to thank my team. I've worked with you for some time now, and I know you're amazing researchers and very humble, modest people, but I know that you're doing phenomenal work, and I want to thank you from the bottom of my heart. It's really, really nice to be working with you, and I'm eternally grateful. Thank you. So there's a lot of progress that we've made, but there's still a lot to do. And I'm not one for like false hope. And the big disappointment for me personally today is that these tubes that were supposed to arrive for study that I'm going to talk about today did not show up. So in the end, we can still do this study. It's about oxytocin and saliva, but yeah, we'll ask you for some patience. So there's a initiative called Combined Brain, and they have different sites, locations all over the US. I think Teresa will probably send out the dates, or you have a website where you can check where they will be next, and they routinely collect various tissue samples, and you can take part in the study. So as Tim and Felix have already talked about, we have a very strong reason to believe there's a dysfunctional oxytocin system. And I want to make sure that we're all on the same page, because what Tim just broke down into, let's say, 10 minutes, this concept is extremely difficult to grasp. So why do we see more oxytocin? And why would we treat the individuals with oxytocin if they already have a lot? So this took us researchers more than half a year to really understand, and we're still not entirely there. And just to make it in the simplest terms possible, think about you're going on a vacation, right? You pack your car with all the luggage and you want to drive out of the garage, but the garage it's just locked. You can't get out. So in order to go somewhere, you need to access the interstate. And what happens is oxytocin is just in the garage, but it's not on the interstate. And when we apply intranasal oxytocin, we're flooding the interstate with carbs. So we have receptors on cells, basically the binding sites of oxytocin, and it's just not reaching there. And coming back to your great question there in the back. So we have no idea about oxytocin receptor up or down regulation. We just don't know that. And we also don't know whether no oxytocin is released at all. I hardly doubt that. Yeah. So without any oxytocin, without any vasopressin, a very related cystopeptide, I think the bodies would not function the way they are. So I think it's like drastically reduced. maybe like 10, 20%. And there's a myriad of questions we still have to answer. So, but I want to make sure that you understand oxytocin gets produced in the cells, but it's not released. It's not acting where it's supposed to be. So I hope this car interstate comparison works. So I want to very briefly go over what has been done in Prader-Willi research. There's a couple of very, very promising studies that have been conducted, but there's also like very, let's say, mixed studies, negative studies. And what it comes down in the end is that we need to treat as early as possible. This is why we somewhat envy you in the US that you have these amazing genetic testing opportunities. So I just heard about a child with Schaaf-Yang syndrome diagnosed at the age of 3 months. That's incredible. Absolutely incredible. Most doctors and endocrinologists in Germany, in Europe, probably don't even know about Schaaf-Yang syndrome. So this is something that the US is going for it. And so we know about oxytocin Prader-Willi research. It can work, and most likely it's most efficient during this critical period for a number of reasons. Something related to receptors, sure, but often we have a critical period, and if like the damage has been done until a certain point, it's like when you build a house with damaged bricks and brittle bricks up to a point, and then you start a treatment. All you can do is like create a scaffold and make sure that basically it doesn't tip over. But if you, at the very foundation of the house, start repairing the bricks, then it's just much, much easier to have a better outcome. Yep. And so just to break this down very, very briefly, Tim and Felix talked about it. What we think is currently happening is that oxytocin is produced but not released, and we are trying to see what happens if we treat rats very early on with supply oxytocin artificially, and we hope that some of the phenotypes that we see in the rat can be ameliorated. Yeah. And how the study that I was hoping to conduct with you today is going to work is that we want your spit. Yeah, we need saliva. And why do we need saliva? Why do we not take the blood? So there's a couple of reasons for that. Oxytocin gets released from cells within the brain, and it enters the liquor, it's called, or brain liquor. It's the artificial cerebrospinal fluid. So it's a liquid that basically surrounds the cells. And this correlates very well with saliva. Yeah, so we have a very strong correlation between the levels found in the saliva and the brain. Not so much for the plasma. It's basically like a random number generator. Those two do not correlate. So this is why we basically are interested in saliva. The big caveat or problem here is that oxytocin gets rapidly degraded. We have a half-life of between 5 and 20 minutes in the blood and the brain, also in the saliva. We have so-called enzymes, peptidases, that rapidly chew up the oxytocin. So for anyone to participate in that study, this whole process of passive drooling, like spitting into the tube, has to be extremely fast, and then we need to freeze it immediately on site. Yeah. So ideally, the whole process takes less than a minute, 30 seconds or so. Yeah. And then we would have shipped these samples to a company that is based in California called Salimetrics, and they specialize in measuring concentrations of various peptides in saliva samples. And in the end, we have a huge data collection. And what I found very interesting is that we have some Schaaf-Yang patients already on oxytocin. I know about Hayden and a few others. And that's, in a sense, it's very bad for us because we want to establish a baseline, right? But on the other hand, we have the amazing opportunity to have 3 groups instead of just 2. So we have a healthy control subject groups. Ideally, those are siblings that are not affected. We have Schaaf-Yang individuals, and we have Schaaf-Yang individuals on oxytocin treatment. And while there will be a huge variation, a variability in the oxytocin levels, that's just natural, yeah. What we hope to achieve is that at some point we have a large enough data set to then say, okay, at baseline, Schaaf-Yang individuals have lower oxytocin levels, and oxytocin therapy maybe normalizes this, yeah? So this would be the best possible finding, yeah? And we're still a long way away from that, and maybe our theory is wrong, but currently we have strong reason to believe that this could be happening, yeah. I will give you very short information about the procedure itself. It's not going to happen today. I'm very sorry for that. So only yesterday I went to the business center and wanted to check for the tubes because I had the FedEx tracking number. I thought, okay, there's no reason to pick it up. It's been delivered, someone signed for it, but turns out it's nowhere to be found. So before you can take part in the study, would have to fill out a consent form, and there's not one fits all. So it's very complicated. So there's one from 0 to 6, there's one from 7 to 11, 11 to 16, and 70 plus. And there's also different forms for siblings versus affected individuals, yeah? But we'll make sure that if it comes to that, you will have the proper form. Trained members from Combined Brain will collect these samples. They have years of experience and could do this way better than we could ever do. It's the passive drool method, so it's just spitting in the tube, rapidly spitting in the tube, and ideally we have like 500 microliters. 150 will do the job, but that's absolutely feasible, yeah? That's not a lot of spit. And the samples will be frozen and shipped to Salametrics. Samples will be fully anonymized, yes? So we have no idea who's what. We just know, okay, the age, and we know basically the genetic condition, right? And ideally, those data could serve for future studies and treatments. And yeah, so it will be very nice for all of us to stay in touch with you, especially the people that are on oxytocin. And although it's anecdotal evidence, we know that it's likely to have a positive effect if you start early. And also there's the placebo effect is incredible. Yeah. So we know from many different studies, if people are doing something and they believe in something, it can have a positive effect. Yeah. So basically those two effects could amplify themselves. So I want to thank you for your attention. We'll be sticking around for a bit longer. And I want to thank everyone involved in all of the studies. And that's just a fraction of the names. I want to thank all our sponsors. I want to thank all the foundations and all amazing collaborators. And most of all, we want to thank you. You are our most valuable resource. And without you, this would have never been possible, obviously. And it's just something very unique we have. Christian, unfortunately, couldn't be here. He's very sad. But we really love these meetings, to see you once a year. And at some point, we would really love to have you in Heidelberg. Thank you so much.